Okay. Um, right before I introduce Dr. Potter, I have the regular ASM housekeeping things to run through. So first, we encourage you to become a member of both ASM National and the branch. If you would like to join the conversation with your colleagues within the society, it's a great way to do that through ASM Connect and talk to peers and catch up with everything that's happening through ASM. One of those items is the annual microbe meeting. It will be held in LA this year, June 19th to the 23rd. So we encourage you to register and travel to California to that meeting. There's also a number of career opportunities and professional development resources within the ASM website, and you can find that at the Careers Professional Development site. There's an ongoing assessment of salaries levels in the microbiology community, and a really nice way to level set and benchmark what you're doing and how other people are being compensated for that around the country. So please take advantage of that survey. And then now to the point of introducing Dr. Potter. So Dr. Potter got his bachelor's degree at UC Berkeley and is a California native, but moved to the Midwest to pursue a graduate degree at Washington University in St. Louis, where he then stayed on for their CPEP fellowship in clinical microbiology. After graduating from that program, he joined Penn and by way CHOP as assistant professor in pathology and laboratory medicine. He's the assistant director of the Infectious Diseases Diagnostic Laboratory at the Children's Hospital of Philadelphia. And he's the director of outbreak investigations in the Center for Microbial Medicine. I think this is like one of the most exciting new roles that have been created. I wish we had one over at HOP, but yeah. But your work, I think, directly influences us. I'll be able to get that. So good luck, and I hope you're really successful. But the idea here is that Robert is taking his skills as a microbiologist, specifically around next-gen sequencing, whole genome sequencing, and advanced molecular diagnostics, and showing us the value of that and the interplay between that sort of research academic standpoint and the clinical impact it can have in a hospital and a laboratory setting. And this, I think, is one of the first examples he's put together since joining CHOP that he's going to show us about. So identifying intra-hospital norovirus G2 transmission. So without further ado, take it away. Thank you.
All right. Thank you, Kyle, for the introduction. Thank you all for being here. Didn't expect that kind of toast surprise, but it really, I think, sets the mood well. So I'm excited to contribute to this amazing branch legacy as a new resident of both Philadelphia and also of CHOP and Penn. So the title is a little bit aspirational. Right now, it's identifying intra-hospital norovirus G2 transmission. But if I'm ever lucky to get invited back, what I hope to talk to you about a year or so from now would be identifying and interrupting intra-hospital norovirus G2 transmission. And this all stems from an unfortunate reality, which is that hospitals can actually be a very dangerous place for patients. They come in with one thing and they are able to, unfortunately, acquire infections in the hospital. So we don't need to bemoan the numbers, but this infographic kind of quantifies a few of the different aspects of hospital-acquired infections. So it shows that if you're admitted to a hospital, you have a 5% chance of contracting an HAI. I think if you were to look into some specific patient criteria, you can see that that chance is going to get a lot of elevated. It really extends the time that patients are staying in the hospital, which both adds additional morbidity, mortality to patient experience, but also then costs the hospitals a lot of money and is inability to then get a bed for somebody else who needs it. And it's actually pretty fascinating to learn how microbes are able to go from a colonized or infected patient to a susceptible patient via a variety of different mechanisms. And I used this same exact figure, which is from like, I think a 2016 paper when I was a second-year grad student. And it's funny that now as a first-year professor, I'm still using this same thing. But instead of talking about bacteria, I'm talking about viruses. But I think this similar trend can be applied to fungal organisms as well. And that's that there's three primary routes of how the organisms can go from a colonized patient to a susceptible patient. They can contaminate medical equipment. That medical equipment, if it's not single use only, may go room for room. And so it could be a vector for contamination. The hands of both staff and visitors, and as CHOP, another additional element we have to think about are the parents, could also be a source of how organisms can go from one patient to another. And then finally, the rooms themselves can serve as vectors, since bacteria, fungi, and certain viruses are really adept at surviving onside these surfaces. And there's a few factors that we like to think about when we're trying to identify what really is an outbreak. And sometimes it's easy. So for instance, if we have disease X, which for this community, let's say disease X is very rare, and it's infecting patients A, B, and C, and D, and they're all on the same floor of the hospital. Then if we see that day 0, patient A is positive via PCR for disease X, day 1 and day 2, patient B, C, and D become positive. This is pretty clearly an outbreak. Since the disease is rare, it's showing up on all these patients that are spatially overlapping. But conversely, we see a lot of this scenario too. Disease Y is really common for this community. Say, for instance, norovirus. Patients A might be on the same floor as B, but then there's some spatial separation. So maybe they're on different floors than C and D. So then if you have the same exact occurrence of events, it becomes a little bit more difficult from an infection prevention standpoint to truthfully say, this is definitely an outbreak or this is not an outbreak. But what's really fortunate is that now in the era of next generation sequencing, we're able to use the whole genome sequence to give us a molecular fingerprint to help determine if organisms are different between these individual patients or if they're all the same. And then with that sequencing information, we can really make the job of IP&C a lot easier by giving them more detailed information on understanding, yes, these infections are all related, or no, these are all completely unrelated. And why norovirus? Well, CHOP has had a long-standing battle of norovirus. This was actually, I want to draw attention mostly to the date of this one, October 2014. So it's been over 10 years of CHOP having popular press about norovirus impacting patient care. This was an outbreak that affected 19 staffers, which sounded like a lot until we're currently dealing with one that's affected, I think, over 20 staffers. So it really not just impacts the patients, but also the healthcare workers who are taking care of those patients. This is a infographic from a local news website that shows the data from a year ago, which was that about 15% of all tests for norovirus were positive in the Northeast corridor of the United States. While I did my training at St. Louis, and I certainly was aware of norovirus, it really was not as big of a scourge. And I think that the kind of high background prevalence of the Northeast makes sense since this is the most industrialized part of the United States. And then finally, if you just Google norovirus, there's an outbreak going on a cruise ship right now where 200 out of 2,000 people all are positive for norovirus. So this was when I was just Googling popular press articles about norovirus at the end of last year, but it's kind of astounding that already I can update this with something else. Norovirus had a really big year in 2025, and that's kind of continued onward into the big year in 2024. That's continued on into the 2025 year. I really was not a virologist before all of this. I did a little bit during COVID, but everybody did. My background is primarily in bacteria. And so norovirus was a really nice virus to get started in studying because it's a single segment. I don't have to worry about influenza with all its multiple segments. It's about 7.5 kb, so a lot smaller than COVID, which is about 30 kb. And it really only has 3 genes. The ORF1 encodes the polymerase, which then does a lot of other immune antagonism functions. Then ORF2 and ORF3 encode 2 different subunits of the viral capsid. And it's this sort of really astoundingly beautiful viral capsid is what makes norovirus such a difficult pathogen from our perspective, because it's so resistant to both disinfection using most cleaning supplies and also desiccation. It's able to survive, if you think viruses are alive, it's able to live on surfaces for weeks at a time while still remaining infectious, which is in contrast to envelope viruses like HIV or HCV, which really, the second they dry out, they're no longer infectious. Norovirus is also super genetically diverse. Shown here is a review article, family tree, encapsulating some of the different genome groups. The naming conventions for norovirus is a little bit annoying, so we'll walk through it in a later stage. But what really the point of this is to show that it's infecting both humans and animals, so you get a lot of genetic diversity because of that zoonosis. We're really just focusing on the norovirus G2, genogroup 2 strains, which can primarily cause disease in humans, but can cause disease in other animals. And G1 disease can also cause, down here at the bottom of the tree, can also cause disease in humans. But for the most part, these large cruise ship or hospital outbreaks that we see with norovirus are caused by genogroup 2. So our goal is to use whole genome sequencing on norovirus G2 samples to aid infection prevention and control measures. Our approach is to use full-length PCR amplification and Oxford nanopore technology for sequencing. And our hypothesis is that we'll identify spatially temporally linked outbreak clusters with less than 10 single nucleotide polymorphisms. When I made these slides a few months ago, I've kept it at 10, but since I've done a little bit more analysis, I've kind of changed that to 5. So purpose of today's talk, we'll keep it at 10, but just know now, I think from this, we're getting additional granularity. It really looks like 5 is a pretty nice cutoff. And all this work really would not be possible without the kind of coalescence of forces that occurred prior to me arriving at CHOP, which was largely that this Center for Microbial Medicine was created, led by Joe Zacular, Ahmed Mustafa, Paul Planet, and Bob Baldessano. And it's sort of this idea of taking next generation sequencing, taking the proximity that CHOP has to patient samples themselves, and then using this information to essentially help create the kind of next era of precision medicine, where we're using next generation sequencing to help improve clinical decision support, the idea of also downstream line creating some sort of a metagenomic test, and eventually also using CHOP as sort of an opportunity for biotherapeutic clinical trials, both in the gut and also in other disease states. The big interest in cirrhosis and plaque psoriasis and other skin diseases. So for the norovirus sequencing workflow, we have 2 major arms of our study. We have the retrospective arm and a prospective arm. Retrospective occurred because I was fortunate that when I started it in the IDDL, there was 6 years worth of norovirus samples that had been collected and frozen and were just waiting to be sequenced. So that was about 500 or so samples. And so that we processed those from July to December-ish. And then prospective samples have occurred about every maybe 2 to 3 times a week. We'll do sequencing for new samples that are fresh from the hospital. And those have occurred pretty routinely now since mid-December, and with the goal of eventually bringing it in-house. So what we're doing is CHOP actually uses a laboratory developed test for our stool viruses. And so the viral RNA is already extracted. We already know that the sample is positive for norovirus. Now we've changed the laboratory workflow where if the lab knows that a sample is positive for norovirus, it goes into a special rack in our -80 where it gets ready to be sequenced. And so for the retrospective, it was costing us about $11 per reaction to extract the RNA. But we're fortunate that for the prospective sequencing, that cost is already eaten into the clinical assay. And then we're using a 2-step cDNA conversion. So since norovirus is an RNA genome, we first convert it into complementary DNA using this QScript kit. And then we're doing a full-length whole genome PCR using this replica HiFi tough mix. The QuantaBio is a company who has worked a lot with the IDDL prior to me arriving. They created this replica tough mix to be used for COVID sequencing, and it is actually capable of amplifying up to 10 kb accurately. So for COVID, they had to do 3 different PCR reactions to tile it. We're fortunate that for norovirus, it's just a single reaction and we get the whole genome. And then we were quantifying the results of the PCR by both running on a gel and then just doing a qubit high sensitivity. But there was really not like a great relationship between what that value was and whether or not the sequencing actually worked. So we kind of stopped that practice. Right now, we are sending out samples overnight for sequencing to Plasmidsaurus. It costs about $20 per reaction, and their turnaround time is truly astonishing, about 12 to 16 hours, pretty consistently. Our goal, once we have a MinION in-house, it is not yet fully operational, but the goal is to then use that MinION for sequencing. And so the turnaround time is going to decrease because the MinIONs, the reads come off instantly, and so it's really nice for analyzing, but then the cost is going to increase. $20 is a pretty sweet deal, and I think it's because they're doing so much sequencing. They can do it at scale. And then since my background is computational biology, I've been analyzing all of the genomic data using the CHOP Red Hat Linux server. And so in total, it costs about $28 per reaction to get one of these genomes, and then we get next day results into the hands of IPNC. And so looking at our cohort, this is the seasonality data. Nothing terribly surprising. It makes sense that mostly we see samples coming in from February, January, and March, and it starts to pick up over December. Not really being familiar with pediatric microbiology, it was pretty interesting to see that about 50% of the samples are coming from toddlers and preschoolers. So I think anybody with young children will understand that makes a lot of sense. And then about 20 equal numbers coming from infants and also from school-age children, while the remainder were coming from the older CHOP population. About 70% of these samples are being tested, ordered on an inpatient floor, 20% from the ED, and 10% from the outpatient. And as I've learned through talking to our clinical colleagues, a provider who's seeing a child in the ED, even if they suspect norovirus, if the child is critically ill and really for the norovirus, are they dehydrated? Do they need IV fluids? The child will be admitted. And then after they're admitted, then they will order the PCR. And so it's not necessarily that anybody who is ordered from an inpatient really is a hospital-acquired infection, since in that scenario, it sounds like would be more from the community. Most of these patients are just single test orders. We do have a subset of our cohort of which got multiple testing, since a lot of kids with immunodeficiencies can be chronically positive for norovirus and continue to shed it for months to years. And so for our analysis, though, we are including these repeat samples. So the first real piece of data is, how does it work? And the answer is, it works all right. It's 80% success rate. So of the samples, we tested about 500 or so, about 464 assembled great on the first round, and 114 failed PCR. And failing means that we still sent something to Plasmidsaurus, but we did not get back either a full-length norovirus genome, or we got back just kind of sequence gobbledygook. And if it was not full-length, we would then repeat it, and with some level of success. And really, this result, I think, is pretty apparent that it is largely driven by the cycle threshold, or CT value, which is a proxy for how much virus is in your sample. So we still had some kind of miraculous successes occurring above 30 CT values, but for the most part, if you're above 30, the odds of you not working are far, far greater than it working. We still will give it the old college try, and we'll do anything for IP&C. We'll give it 2 attempts before we say, probably not going to work. And then we sort of don't have a great explanation for what's going on down here. These samples have very low CT values, so there should be a lot of virus. Our kind of conjecture is that since that second step reaction needs full length amplification, even if you have a lot of virus, RNA is a very finicky macromolecule. And so if you have sheared RNA or degraded RNA, you're not going to get that full length amplification. And since those primers sit at either end, you're not really going to get anything. And so you could, in theory, design primers that maybe are occurring inside of the genome to get a bit more amplification and then follow that up with Illumina sequencing. We have not tried that. We were really happy, though, that we didn't think of this idea. This came from a great paper from Public Health Canada, where we got both the primer sequence, the idea to use nanopore, but the sort of major innovation that we tacked on was that rather than using more expensive in vitrogen units or reagents, we use samples from QuantaBio. And so in that original paper, they tested about 57 samples and had a 70% success rate. We've tested now about 10 times more, and we have an 80% success rate. And really the biggest difference is that our protocol takes far, far quicker, mostly because for the long PCR step using the Invitrogen products, it's like 1 minute per kilobase for amplification. And so for a 7.5 kb virus, that means you're going to be amplifying for a lot. But whatever their QuantaBio is doing to mutate their Taq polymerase, it's able to move much, much faster. And so the long PCR step only takes an hour. And so that means really that our kind of pie in the sky dream of a same day turnaround time of kid gets positive norovirus PCR in the morning, end of the day, we have a genome sequence, I think is actually feasible. So now just to go a bit over into our computational pipeline, we kind of have this order of proceeding from least granularity to most granular. And least granular is genotyping, where we're giving just kind of a large level overview on the phylogeny of the norovirus, kind of analogous to like MLST type for bacteria. Intermediate in that is a maximum likelihood phylogenetic tree, where we can take all the noroviruses, we can align them together, and we can look to see at the tree. And then the most nitty gritty is temporal spatial SNP analysis, where we're fortunate that we have a pretty big clinical informatic metadata pool through our IRB. And then with the whole genome sequence data, we're able to exactly say where every patient sample was, what room it was in, and how related they are to one another. And in the interest of time, really focus on those most granular and the least granular. So the first thing we wanted to do is just to kind of QC our data to make sure everything looks right. So norovirus should be about 7,500 base pairs. And we're fortunate that it's kind of exactly what we see in our cohort. Our average was 7,568 with a standard deviation of 47 nucleotides. And so these two red bars show the mean plus or minus 1 standard deviation. And you can see that we really get most of our samples occurring in that expected size distribution. We had a few that are a little bit shorter and a few that are a little bit longer. And the longer ones when you blast them, they actually come back as being related to another large noroviruses. And so we're really not sure, kind of, you expect there to be some heterogeneity. The shorter ones look like they maybe just didn't have like a part of their genome amplify. So they kind of get removed from the other part of the downstream analysis. So now moving to the genotyping, there's a really annoying scheme to learn at first. So there's a few numbers and a lot of words to go over. So first is we take norovirus, we know it's a norovirus, and then we can look at the polymerase gene and the capsid gene. The capsid gene gives us the first number that I've said a bunch of times, which is G2. And so this is a genogroup. The genogroups are the G1 through G10. They're set using Roman numerals. G1, G2, cause disease in humans. G4, a little bit. The other one's more animal disease. And they also will give you an additional genotype. So this is G2, 4, and then Sydney. They give you a location based off of where it was really first discovered. And now the polymerase gives you additional information. It's always denoted with this little P, and there's just another number associated with it. So when you put it all together, that gives you this kind of word salad like G2.4 Sydney P16 strain, which is the most common genotype that we see at CHOP. We see some of these other strains, G3 P12, G17 P17, and in total, we actually identified 30 different genotypes from this assay. But really, these big 7 make up 85% of the cohort, and so they're really, for the most part, the ones that we're interested in looking from a infection prevention standpoint. And since I really had no idea about anything in norovirus, when I started this project, just asking Dr. Google, brought up these papers. And so I was fortunate to see that for the Sydney strain, that G2P16 Sydney, is generally the globally dominant norovirus. And it emerged from Australia in 2012 and then kind of quickly took over the world. So it makes sense that what we see most commonly at CHOP is what we see most commonly globally. It was really interesting that then when we took that genotype data and we looked at it temporally, we can see that the Sydney strain, so shown in red, is for the most part relatively stable throughout the change of the seasons at CHOP. It varies between 25 to about 43% annually. But what was really interesting is that we also see kind of the opposite. So we see temporal dominance of the G2.4 Sydney P16 strain, but then we can see this kind of blue guy, the G17 P17, had a really big year in 2024, and that's continued into 2025. And for the most part was kind of present through these other years, but really did not take up the lion's share of genogroup or genotype proportionally until last year. And we see the opposite. So this G6P7 had a really big year during COVID. Of course, our numbers are a bit lower because people were just generally getting less gastrointestinal disease. But then post-COVID it's kind of withered away, but still stayed somewhat present. So it was interesting to see that this was important because I ended up writing a vaccine grant to the NIH. We'll see how that plays these days. But it shows at least that when you're formulating a vaccine, you really need to consider not only the kind of dominant Sydney strain, but some of these other ones should also get considered inside of that formulation. And for the SNP analysis, so really it's very simple what we're doing. We have 2 different genomes, we're aligning them, and we're just counting how many SNPs or single nucleotide polymorphisms exist. But it gets more complicated. This is example for 2. Since we have 500-ish genomes, 500 pairwise interactions give you a matrix of 124,750 pairwise comparisons, which is too big of an Excel sheet to really look at it and get anything useful. What we can do is you can take that matrix and you can distill it down into a histogram. And so what we're looking at in this histogram are the pairwise SNP distances between every possible pairwise interaction within our cohort. And then we're counting the number of values related to that SNP information. So it was interesting to see like a lot of these different peaks. And I'm still kind of working into like, what do these different peaks correspond to? You'd imagine that over here, you essentially get the 2 opposite ends of the phylogenetic tree having the maximum distance from one another, and then some of the distances between the different genotypes. But from an outbreak investigation standpoint, fortunately, none of that matters. We don't really care. All we care about are these genomes in this bucket. So this bucket is indicating the strains that are most closely related to one another. And then when we zoom in on just these strains and we look at them as a network diagram, we get this. So this is a network diagram figure where each of these black dots represents a single genome. The gray lines connecting the black dots indicate that there is less than or 10 SNPs between one another, which during this time is what we considered as a cutoff for clonal relatedness. And when we do that, we can see that there's 30 pairs, 14 triplets, 6 quartets, and 7 clusters containing 5 or more genomes in each cluster. So we're just going to dig deeper into these larger clusters. And when I saw this and I looked into the patient metadata, I was really happy to see that thankfully cluster identity essentially correspond exactly with the temporal time of collection, which makes sense that these outbreaks are going to be linked both in time and also spatial identity. So these kind of generally occurred over the winter months, as you'd expect. The biggest one actually occurred in February 2024, I think like right while I was interviewing at CHOP. And then there's a few from the previous years that when I was talking to the IPC folk, very much remember exactly like what December 22 was like for norovirus. And then when we overlay the genotype information, it makes a lot of sense that since that Sydney strain is the most common one we see at CHOP, it is responsible for 3 out of 5 of these different outbreak strains. This G2.4 Sydney P4. So now that we know, now we're all experts in norovirus genotyping, we can know that this is actually the same capsid as the Sydney strain over here, but they have different polymerases. And so that one was also able to cause an outbreak in January 2022. And then this December outbreak was the G3P12. So since this was the biggest outbreak, this was kind of the one that was most exciting to look into a little bit further. It compromised 16 genomes in multiple hospital floors over several weeks. Now, Kyle and I were at a meeting in January where I heard the term radiculogram for the first time to describe kind of really complicated looking genomics figures. And now I really like that term. So this is our radiculogram. It's a force-directed network diagram. So basically, each of these individual black dots represents one of those norovirus genomes. The numbers correspond to our de-identified nomenclature scheme. So these are all from our R or retrospective cohort. And then it's force-directed, meaning I give Cytoscape the network diagram, and then I say, give me the maximum possible separation between all these individual points. And it came up with this scheme. And then I colored the edges to correspond to the SNP distance. So if you see a red line, that means there's no SNPs. These are exactly identical to one another. Blue lines mean there's one SNP. And when we take the same strain and we sequence it a bunch of times, the error of the assay is about like 1 SNP. And then 2 to 10 SNPs are these gray lines. So what I hope is really apparent is that you can kind of see in the red that there's this group up top of 5 genomes that all are, for the most part, exactly the same. There's this triad down here of 3 guys that are all the same. And there's 2 up here with a lot of blue in between them, indicating that these are very, very closely related genomes. Think the maximum genetic distance in here is 10 SNPs. And then we can create a time-resolved phylogeny. And so if we were to just do normal maximum likelihood phylogenetic tree, anything that's identical just gets a vertical line because the program can't tell it apart. It's identical. But where this was done using tree time, and tree time, you're able to then feed in the timestamp as metadata. And so it's able to give you additional differentiation in that branch length. So all of the genomes that you're going to see in these red boxes have 0 SNPs between the other genomes in that red box. So it was interesting to see that that 5 group cluster up here, that is all of these genomes. And so they're all exactly the same from a sequence standpoint. They all are occurring in ICU-A, which I'm de-identifying for this purpose. But we see that strains from ICU-A throughout the phylogenetic tree in 2 other locations. And this outbreak was not really confined to just ICU-A. We can see additional spread to ICU-B. And again, even though it's a phylogenetic tree, there's only 1 SNP difference really between this strain and these other strains, and additionally 1 SNP between here and here. And we can see the same exact virus. So again, these are 0 SNP distance, but this one's occurring day 30 of the outbreak in ICU-B. This one's occurring day 15 in ICU-C. So it was just truly remarkable to see the level of temporal and spatial overlap that these norovirus are able to cause. It was also really interesting to see that 2 of these elements were from the emergency department. And so the the like early beginning slide on disease X and disease Y is sort of how I'm trying to think about how do we determine if a disease comes from community or it comes from the hospital. Obviously, disease will have originated in the community, but it's only a matter of time before it spills over into the hospital. And so the fact that this particular R437 is coming from the ED, it's also the most distantly related norovirus genome within this phylogenetic tree, since they only share a common ancestor here. But this 4561 really looks embedded within it. So to me, that indicates that there is some level of circulation of this particular strain of norovirus occurring in the community. And so this likely represents some sort of a spillover event, but that this actually is bona fide transmission inside of the hospital. And the big kind of gap that I really wish I could get access to is both the workers and also the parents, because it's really nice to get some of those samples. Because if I was able in public health, and you can do really high detailed contact tracing, it's really fascinating to see, is there any sort of social contact between kids or family members within this group? And so that was an example just to show the large cluster up here, but we can take this concept and we can apply it for all the other samples in the genome cohort. And I've started doing this. I don't have the data showed quite yet, but what we've basically been doing is taking each of these black dots and now reassigning a color if it came from inpatient, outpatient, or ED, so that we can more visually see and to show to IPNC, look, we are getting kind of sustained transmission that starts in the ED and then moves into the hospital. And so that was all from the retrospective stuff. From the prospective stuff, we've been sequencing. We have about 80-ish genomes so far assembled. When we do the same approach, we look to see who's exactly similar to one another in a 0-1 SNP distance. We come up with here. So this was a big cluster of disease that occurred a few months ago at CHOP that thankfully did not spread to any patients outside of the unit. And then these are two examples of just kind of singleton infection cases. And we're going to dig into this one in particular because it was kind of, I think, really drills down on the point of like, why is doing this important? So this was a story of patient A initially presenting to the ED with gastroenteritis. This was occurring end of December, beginning of January. So when you expect there to be a lot of gastroenteritis, a lot of community transmission of norovirus, patient A is really not that medically complicated. I forget how old they are, but it's kind of just kid, stomach pain, comes in sick. They order the norovirus PCR, which again is this sort of multiplex test that we look for a lot of other viruses. And then patient A, since they were not medically complicated, they were not super dehydrated, they got discharged. Immediately after patient A left, patient B, who is very medically complicated, and ironically was presenting to the ED for an infection-related issue because they have a JG tube site infection that the IDDL ended up culturing Pseudomonas aeruginosa, occupies the same exact room that patient A was just in. The next day, patient A result comes back as positive. Patient B, because they're medically discharged, or sorry, because they were medically complicated, and because we were very concerned about that Pseudomonas site infection, patient B was admitted. 3 days later, patient B develops gastroenteritis inside of the hospital. The norovirus PCR is ordered, and that same day comes back as positive. The next day, we perform our sequencing assay on day 4. Day 5, we get results back, and we can see that there's 0 SNPs between the sample on day 0 from patient A and the sample on day 3 from patient B. We email this. We have a few contacts in IPNC who, when we get suspicious results like this, we email them. And it took our colleague in IPNC 20 minutes to figure out that patient A and patient B overlapped in the ED by exactly that room. And so thankfully with this result, there really was no ambiguity. We have a few cases now where there's a 3 to 4 SNP distance between related clones. And so that's a little bit harder to say. I think that as we get more sequencing, then we can really do some better time-stamped phylogeny to figure out what's the actual molecular evolutionary rate of norovirus. I was kind of skeptical of molecular clock stuff as a grad student, and I still don't know how I feel about it now as a professor. And with the goal being, like, how can we use this information to create actionable things the hospital can do to stop this? And so the hospital does have a norovirus-specific cleaning protocol. It's just very labor-intensive. They actually have to call the environmental services. They have to get kind of the special disinfectant. And so they're able to do it. It's just a lot of hoops that needs to be jumped through. So the ED did nothing wrong because at this point, they did not know that that kid had norovirus. But maybe it means that we should the ED use the norovirus protocol for all cases of gastroenteritis during the winter? Just because this is an example for norovirus, we're not sequencing rotavirus, we're not sequencing astrovirus, but you very well could imagine that that scenario could be replicated by these other viruses. And so although it might be more labor intensive and expensive to do that high level cleaning, it could contribute to lowering our patient mortality. Morbidity.
Is it also could be the same staff that will receive patients? So is it environmental?
Yeah, that's another good question. It could definitely, we're blaming the room. We're blaming like maybe the physical like table where the kid sat down on, but yeah, very well could be staff. The other thing that makes norovirus a big deal from an infection prevention standpoint is that it does not, hand-based alcohol sanitizers are not effective at all against norovirus. You really need the soap and water, and it's mostly actually they're not even killing the norovirus. It's just the soap allows it to be kind of loft off your skin and go down into the sink. And so now that as we're kind of dealing with another outbreak, we're making sure to educate the staff that you cannot use alcohol-based sanitizers. You need to actually physically wash your hands to try and get rid of it. So in conclusion, shown that whole genome sequencing of norovirus is cost-effective, relatively quick. We have an 80% success rate on the first attempt, strong dependence on CT value. That Sydney strain is the dominant genotype we see at CHOP, other genotypes varying annually. And the SNP distance really helps show the clusters of highly related genomes spreading within the hospital. Currently, we continue to do sequencing prospective samples for IPNC efforts. I think we did 9 samples today, and we did another run on Thursday. So there's been 9 new positives from Friday, Saturday, and Sunday. We're using the NGS data to target IPC interventions, so hand hygiene reminders, bleaching rooms. Right now, it is being done by me and then the 3 kind of R&D techs that the IDDL has, but we are in the process of codifying it into our normal molecular SOP. And so then it's going to be something that just when they're on the molecular shift, they do the norovirus PCR, they put it in a special rack, and then we just go drop it off in the box, or we do the sequencing in-house. We have the MinION in our possession. We had to buy like a separate gaming laptop because the computational energy of the MinION was so intense. And we wanted it to be kind of not super on the CHOP website so that we can get access to all the MinION data. And then eventually my pie in the sky dream is to validate it in a CLIA approved manner so that the results can interface with the LIS and that we can kind of get that reported out in the electronic medical record. CLIA and sequencing is sort of a topic that's beginning to merge together. So a few institutions like Wadsworth in New York has a CLIA validated sequencing assay for carbapenemase detection. Virginia has a CLIA validated sequencing assay for looking at clonal relatedness. We're right now operating outside of CLIA because we're saying this is not used for individual patient care. This is being used for epidemiology and infection prevention purposes. But my goal would be to make it CLIA approved. And then CDC has a CaliciNet surveillance network. So there's hundreds of thousands of genomes of Shigella and Salmonella online because those are required to be sent to your state and local public health lab for typing. There's a huge lack of norovirus and the other gastrointestinal viruses because you're not required to send it. CaliciNet exists, but it's kind of a volunteer organization, and it's kind of opaque from the CDC website on what actually you need to do to get invited to join CaliciNet. And I'm also not sure would be able to because we're doing whole genome sequencing, and this was all built on the premise of this smaller amplicon-based approach. So if I can't upload it to the CDC directly, I'll still just submit him to NCBI Sequence Read Archive, and then anybody will have access to him. And I would not be up here talking to you all about our exciting data without a lot of people who have been contributing to this. So most notably, my co-directors, Dr. Rebecca Harris, who's our director, and KP Smith, who is assistant director, our lab manager, Kara, a supervisor, Jackie. Everybody in the lab has been super helpful in our R&D team, Jess, Jeff, and Mike. This is all occurring through our pathogen registry, IRB, largely through Paul Planet, Ahmed Mustafa, and Joe Zakular at the Center for Microbial Medicine. And then our partners in IPNC, Erica Hayes, Logan Grime, and Heewon Yoon. And that is it. So if there are any questions, I'd be happy to answer them in the remaining time.
Here.
have the microphone.
Sorry.
Next, I'll start with the first question is, when thinking about implementing these things, like the genomics is complicated enough, but then getting infection prevention to identify a potential link and then come up with some strategy to act upon it is like a whole new frontier for them too, right? And it looks like you're showing that there's norovirus that's highly related, that might stand 24, 28 days. But what is have you modeled, like, if intervening this quickly, how many infections could we prevent?
Yeah.
And
Yeah.
what do you intervene on? Because it's like, there's already hand hygiene.
Yep.
That's like really, really great attention to. How are they going to get better at that hand hygiene stuff?
Yeah, and there's no, like, for Staph aureus, there's decolonization, but there's really not, you can't decolonize for norovirus, and it's also one that thankfully we're not, like, we don't think that there's any kind of asymptomatic colonization going on. Norovirus tends to make itself known. But yeah, that is the kind of question that we are dealing. That's why I did this example, where it's very clear that we had a linkage, and there's an actual item we can do. This other case, we're really not sure, where there is like a patient who was in one wing of the hospital, and then a week later, that same exact norovirus, 0 SNPs, was in a completely separate wing of the hospital. And we don't know, like, the IPC has been looking to find, is there any common providers, is there any common equipment, but there's no clear linkage. And so, yeah, we're still learning how to take this data. And really, when I was very new and talking to the IPC people, I learned mostly that they tend to think about the hospitals like vertically, or horizontally, in terms of like certain individuals will cover certain floors, and rather than taking it mostly from a pure pathogen standpoint. And so at least now we're kind of combining all that together. And so the IPC person who covers like the 3rd cancer ward would have to interface with the IPC person who covers the like orthopedic floor. And in terms of interventions, yeah, we're really not sure. One thing I do want to do, which I think might help, is that I want to kind of create like a response program where as soon as a kid becomes positive for norovirus, we're alerted and we can immediately, with IRB approval, go in, swab as many high contact surfaces as we can, maybe consent parents or the guardians to get swabbed, so then we can see kind of more established where the directionality is kind of coming from. And that might help then figure out what would be the best interventions.
Very nice talk. I was just wondering, I used Minion many years ago at its early ages.
Oh.
And quite a few mistakes.
Yeah.
So I was wondering, I saw you did some repeats in the first slide. How good is it now for 7.5?
Yeah, it is crazy.
Check a little bit these repeats, were they consistent?
The repeats of the genome, or the repeats of like repeating a sample and then testing it again?
Yes.
Okay, yeah, we've tested it. Yeah. So norovirus is interesting. The genome has a ton of repeats, where it basically starts with just it has just this long sequence polyadenylation at the end, and which then makes the sequencing actually easy, because this is really nice to just have very specific primers to bind to. My graduate school lab got one of the early MinIONs in 2015 when they first came out, and it was garbage. We called it a random letter generator. But then thankfully, it's crazy how much changes in 10 years. Now the chemistry is way better. Because the error is random, in theory, it should be better than Illumina if you just kind of sequence to a multiple depth. And we, although acknowledging that, like, by doing this kind of full length PCR amplification, we might get errors due to the Taq polymerase rather than if we were doing like a tiling approach. But since for what we're trying to do is just compare our own norovirus, we really don't care too much about like globally what is norovirus doing. At least all the errors should be mitigated because we're all processing them the same way. But that is also why in our kind of test set, we did take the same thing and we just sequenced it a few different times to learn, like, for the most part, they come back either identical or like a 1 SNP difference, really. And then, and then that was just for the virus part. For the chemistry of the nanopore technology, it's amazing how much has changed. And now, it's very good.
You just have to have relatively few changes from patient to A to B, etc. So you really have to be very exact.
Yep.
To be able to follow.
Yeah, and that most of these changes are occurring and not causing an amino acid change too, and so we don't have the protein sequence to also then say, okay, this corresponds to, if we see the change in the nucleotide sequence, it's helpful to then see change in the protein.
It's one more question. Do you think you could actually focus on some regions instead of doing the whole genome?
Yeah, you could definitely do that. So the current typing scheme that like CaliciNet uses, I think, is like a 600 base pair amplification sequence that covers like this region, which both captures the capsid and then also the polymerase needed to do the dual typing strategy.
Because of vaccines, you know, focus on.
Very interesting thought. So, I'm just curious about the mutation rate in selective mutation rates. It doesn't seem like it's changing a lot, but if you were to look at the samples with the repeated from the same patient, have you seen any post-viral interactions that have changed there?
Yeah, I was really interested in doing that, and that's also kind of why I have my mindset for this, that it's the transmission story, not an evolutionary story. But as like a basic science biologist, when you get all the sequence data, it's like we have to learn to see like what is this population really doing. And so to get at the evolutionary question, I took from the literature, there's not a lot on whole genome sequencing, but there's a lot on amplicon sequencing. And then the amplicon people will then kind of use like 0 to 2 SNPs for that 600 base pair section. I forget exactly the length of it, but basically then when I scaled up, when I took that 2 SNPs for that smaller amplicon section and I applied it to the full length of the norovirus genome, it came up with something like 34 SNPs, I think was the cutoff if you use that rule, which then was a good place to start, but clearly seems too large relative to kind of the differences that we're seeing. And so what I really want to do, since I do have this very nice timestamp data, because we're very fortunate that the Epic Health Interface just makes it a lot easy to do that informatic query. That in grad school, I was never really able to do like Bayesian phylogenetic dynamics because we didn't have that really good timestamp. So I wanted to take is use Beast2 to then do like molecular clock stuff to then see, can we, now that we have several hundred genomes from all the different parts of the norovirus tree, we have good timestamp data, can we use this to then get a more accurate representation of like what the molecular evolution rate is? Because the other thing that I've been thinking about but haven't explicitly tested yet is that like right now I'm applying the same rate of evolution for all of these different genotypes. But you can imagine, especially if you're a virus that has mutations in its polymerase, maybe it's replicating or maybe it's accruing mutations at a far different rate than its sister clade like down here. So yeah, that's something that we're probably not going to include kind of in this first pass analysis, but with this cohort, I think would really be adept to address.
And the second question is, can you predict from the genotype the virulence of changing disease progression to mutation?
Yeah, that is another great question. And that work all comes from Kim Green, who's a really phenomenal norovirus scientist at the NIH. I think has been there for like 20 or 30 or so years. Has actually done a great, very recent paper where they took a lot of these different genotypes, then they infected organoids. So a big issue with norovirus is that it cannot grow in mammalian cell culture, but they fortunately learned that organoids actually are capable of allowing it to grow. And so then when they take some of these different strains, they found that the Sydney strain had sort of much quicker replication time inside the organoids compared to these other strains. And I don't know for organoid biology, like how much batch variation you get between the different lines. But there in that paper, they then indicate that, this faster replication time could explain one of the reasons why that Sydney strain is the sort of globally dominant one. Because you'd expect norovirus very rarely kills, but there must be something that then has allowed the Sydney strain to be so good at causing infections around the world. The tapping voice.
Yeah, I have another question about the repeated samples on the same sequence. I'm wondering if you were able to correlate some of your failed PCRs with some like short-term shedding, and then if there is any current data on the infectiousness of patients that are shedding Pfizer, or the as infectious as those that are.
Yes, you hit the nail on the head, which is basically that for some of these kids who we see like 4, and then the max that we had in this analysis was 4, but now I've been redoing the data for the paper, and we actually have some that got, we have 3 kids who got tested 5 times. And for the first 3 samples, the CT values are like really down here. But then in all 3 of those, the last CT values are like 33 or 34, 35 above. And so that would indicate that, yeah, probably some of what the loss we're seeing is just the kids are improving. And so there's less norovirus in their stool. And so we're seeing failing of the PCR because there's less virus possible. And we also see some instances where some of these genetically identical viruses, when we look, they're like, oh, wow, these samples are occurring at the exact same time. And that's because some of these interactions from our retrospective cohort are like siblings who all are presenting to the ED at the exact same time. And so we didn't sequence their viruses multiple times, but it was really assuring to see like, okay, 2 kids from the same household have the exact same virus. So it's another good kind of like orthogonal way to show that, all right, we can trust our sequence data.
I was just gonna ask if you could use this to the outbreak or the contamination event.
Yeah, we're really interested. We're not doing that we haven't seen that for noro yet, thankfully, because we haven't had a lot of contamination. But CHOP does breast milk culture for new mothers, and we are using the sequence data because we've had like a run of bacillus samples that have all been positive. And of course, from the lab, there's hundreds of bacillus species. They all look the exact same. MALDI can't really help you beyond that. But with the sequencing, we were able to actually show that the bacilli each correspond to the batch of breast milk. And so, because initially it was kind of like, ooh, is the lab contaminating it? Because, you know, bacillus is everywhere. It's on everybody's skin. It's in a lot of our like packaging. So at least we're able to use the sequence data to show like this is not coming from the laboratory. The breast milk bank still is not sure exactly like where is all this bacillus coming from. But we at least shifted the blame away from us. But it's not something for the norovirus. We haven't seen it causing contamination, thankfully. But you know, it's always a concern because especially some of these viruses have very low CT values, so they're really just they could be chock full everywhere.
I was just wondering, do these noroviruses make a lot of defective viruses?
I don't know. That's a really good question, and CHOP would be the best place to answer that. CHOP/WashU, ironically, with Carolina Lopez, formerly of both Penn, and then now being at WashU, my former institution. And then like Shon Gupta, who's continued to do a lot of great work in that area. I really don't know enough virology to know about if norovirus is doing that, but it is definitely something that could be for us. Mm-hmm. Yeah, like that could be it.
Where you going next? Like, this technology seems like it would adapt really well towards like enteroviruses, something else transmissible and single-stranded RNA virus.
Yeah, I wish I had a slide on that because we're always thinking about what's next. And so what's next is we basically want a single assay to rule them all. And so instead of a norovirus specific PCR, we're working right now on a pan-virus metatranscriptome assay to basically say like, because it seemed like every other week IPC might say like, we have a rhinovirus outbreak, we have a metapneumovirus outbreak, we have an RSV outbreak. And norovirus was nice to get started because the genome is just very amenable to doing that targeted PCR. The diversity within rhinovirus is probably greater than the diversity of some organisms as a whole. And I don't even know if you could have a single assay. I've not seen a paper that has like a single assay that will detect and amplify the full genome for like all the different possible rhinoviruses. And so we're really fortunate that we were able to hire a research technician a few weeks ago who came to us from Pitt, where she was also working in a virology sequencing laboratory. And in collaboration with QuantaBio, the company who's made these reagents, they have a single kind of assay that will take you from extracted product all the way up to Illumina sequencing libraries. And then all you need to do is just pull your libraries together. And so we've essentially, during this most recent respiratory season, we've been saving all double positives. So like flu A and RSV, flu B and COVID. And I think we have 48 of them. And so we're using that as kind of our test set to say, okay, Cepheid says there's 2 viruses in this assay. Can we assemble the genomes of both of these viruses? And then that shows us that like if something pops up, then we have the ability to sequence it. And I thought I was being really creative and cunning using metatranscriptomics for sequencing, but UCSF Charles Chu laboratory published a paper doing that exact same stuff a month ago. So at least it then shows up. Look, now hospitals are very competitive with one another, so it's easy to say like UCSF's doing it, why shouldn't we? But yeah, that's kind of where we're going for the future. In addition to sequencing bacteria routinely, and then if anybody here is a fungal biologist, I've been trying to do mold genomic DNA extractions, and there's tough nuts to crack. but I want to sequence them really bad. So yeah, that's kind of where we're attacking virus, bacteria, and fungi.
Right. Thanks again.
All right, thank you, everyone.